for taking the time to join us today. For those of you who aren't familiar with us, Inscopix is a neuroscience tech company based in Palo Alto, California, the heart of Silicon Valley, and we provide complete miniature microscope solutions to scientists that want to map brain circuits in freely behaving animals. We're super excited to have Cody Siciliano join us today. Cody is currently an assistant professor at pharmacology at Vanderbilt University in Nashville, Tennessee, and he's part of the Vanderbilt Center for Addiction Research. Cody has had a long career interest in addiction ever since he first received his PhD in neuroscience from the Wake Forest School of Medicine in Sarah Jones's lab, and he continued his exploration of neural circuits underlying addictive behaviors during his postdoctoral fellowship at MIT with Kay Tai. Cody now runs a vibrant research lab at Vanderbilt, and we're thrilled to have him with us today. So before we get started, I want to make sure everyone attending understands the rules of engagement. We will ask you to please provide your questions to us via the chat window that is located down at the bottom of your window. And we will do our best to prioritize those questions so that Cody can answer them at the end of this presentation. And please know that we may be able to not address everything that is being asked, and it will depend on the time that's left and available. So without further ado, I'm going to turn this over to Cody. Cody, thanks again for joining us today. It's wonderful to have you here, and I'll let you navigate the screen share, and I will turn off my microphone so that you can begin talking.
Okay, great. Thank you for that introduction, and thanks everyone for tuning in to hear this. This is my first time doing one of these on Zoom, so hopefully everyone can see my slides here okay. Hopefully everyone can see me okay too. This is my first time not wearing pajamas in like 3 weeks, so I'm really hoping to get as most mileage as I can out of getting dressed in real clothes. So as she mentioned, I'm a new assistant professor at Vanderbilt University, so I just started my lab about 6 months ago and then had to shut it back down again. So I'm actually going to be mostly showing you data that was collected during the end of my postdoc in Kay Tai's lab at MIT. And so I'll try to talk a little bit about what the lab is generally interested in, how we're applying some optical tools, including endoscopic scopes, to address these questions. Then I'll show you one specific example of sort of how we approach the biological question with these, and then wrap up with some future directions of what we're doing in my own lab at the end. So one thing that's really been a driving question throughout my career is trying to understand the neurobiological mechanisms underlying alcohol use disorder. And one of the things that's really most fascinating to me about this question is the really stark individual differences that you see with this. So as you probably know, at least in Western culture, exposure to alcohol at some point during your life is almost ubiquitous, but it's only a relatively small but very persistent subset of people that will go on to develop an alcohol use disorder. And so one of the major questions that we try to address in the lab is that how is it that so many people can have the same stimulus, alcohol, and have these wildly different outcomes? How can we understand the neurobiology of this, and ultimately how can we intervene? There's lots of different aspects to an alcohol use disorder. One thing that we know that's really important is changes in the way that aversive stimuli are processed. And so one of the most famous theories in the field, and probably the most well-studied, is this allostatic or Kubian model, where you have these really slow changes in aversive processing. So over long exposure to alcohol, you have these changes in the allostatic state, whereby you have sort of negative emotional processes that increase over time, and eventually you get to the point where you're actually using alcohol through a negative reinforcement mechanism. So in other words, to alleviate the symptoms of withdrawal rather than for the positive aspects of alcohol. And so like I said, this is known to be super important and is really well studied, but we also know that there's changes in the way that acute aversive stimuli are processed. Drinkers are actually less sensitive to aversive outcomes that can happen as a result of alcohol drinking. And we can conceptualize this as compulsive drinking. So this word compulsive, people like to argue about its exact definition sometime. I'm going to use it a lot, so I'm just going to operationally define this up front. So when I say compulsive drinking, I mean continued alcohol use despite a negative consequence associated with that action. So you can Sort of think of this in a very simple way. So in an adaptive animal, you'll have some baseline level of alcohol consumption. If that action is paired with a punishment, alcohol consumption will decrease, whereas in a compulsive animal, the alcohol consumption will be relatively unaffected. And so we certainly are not the first group to look into this. So there's really a mountain of evidence that implicates plasticity in medial prefrontal cortex in mediating the development of these compulsive drinking behaviors. So just a few examples here. So in human alcohol use disorder patients, we know that cortical activity evoked by alcohol-related cues correlates with alcohol craving. And another way that you can kind of think about compulsive drinking is a deficit in top-down control of response inhibition. And the first thing you think about for response inhibition usually is the medial prefrontal cortex. And there's, again, a lot of literature that implicates this region in mediating this type of behavior. And so one of the ways that medial prefrontal cortex can mediate this sort of general behavior in many different contexts is through its really dense interconnectivity with sensory regions, and then it's thought to drive this top-down control through its subcortical connections. And so while this is what makes the region so interesting, it can also make it difficult to study because there's so many different circuits that it can be difficult to parse what's going on. So in the preclinical drug abuse and alcohol literature, again, it's been pretty well established that there is plasticity in prefrontal cortex, and that relates to excessive drug use and compulsive drug use. But a lot of it, what's been done in the field is to sort of look at plasticity after long-term exposure, and a lot of this has not been able to parse apart the specific circuits that are involved. So those are two things that we kind of wanted to address here. One is how is aversive information being routed through medial prefrontal cortex, and how does that change with alcohol exposure in vivo? And two, what are the specific circuits involved here? And so I'm not going to go through all of the preliminary data that kind of got us to this point, but we really got interested in this projection from the medial prefrontal cortex into the periaqueductal gray. And so the PAG has been, at the point that we started this study, the PAG, of course, had been studied extensively and implicated in aversive processing. But at the time when we started this, again, this projection from medial prefrontal cortex into the periaqueductal gray had not been investigated yet. And we reasoned that, if the PAG is mediating responses to aversive outcomes, and the medial prefrontal cortex is sort of exerting this top-down control over your response to those outcomes, if this circuit was dysregulated by alcohol exposure, that could be a way that alcoholics become less sensitive to these aversive outcomes. So to address these questions, we of course needed a way to observe the activity of these neurons in vivo. So for that, we turned to this deep brain projection-specific calcium imaging, so Inscopics in this case. So we injected pre-dependent vectors into medial prefrontal cortex carrying the calcium indicator GCAMP6, and then a retrograde virus carrying Cre into the dorsal columns of the periaqueductal gray. So this gives us projection-specific recombination of the indicator. So I'm going to refer to this circuit throughout the talk as the MPFC to PAG. I'm not going to say dorsal every single time, but all of the viral injections into the PAG were restricted to the dorsal columns, and in the prefrontal cortex, we're in medial prefrontal cortex and collapsing. We have expression in both IL and PL cortex. So this gives us, like I said, projection-specific expression of the indicator. We can then implant one of these Grin lenses over medial prefrontal cortex, and that gives us chronic optical access. And then, of course, as you guys know, using these Inscopics miniaturized microscopes, which we can attach to the animal's head just prior to the recording session, we can then observe the activity of these projection-defined neurons in vivo during free behavior. So this is what the data look like. So you'll see cells in the field of view. Increased brightness is indicative of increased action potential activity. And so we started, this was actually quite some time ago. I think this was actually my first in vivo optical recording. And so we were just getting this going at the time in Kay Tai's lab. And so we started out with asking a really, really basic behavioral question. We just said, do these neurons respond to an aversive stimulus? So to do that, we just put the animal in a conditioning chamber and then delivered unsignaled foot shocks to the animal. So you'll see a representative video here with a couple seconds of baseline. And then when the foot shock is delivered, you'll see a red square around the video. And so you can see we just get this really sharp and then prolonged activity in response to the foot shock. Like I said, this video was a while ago. It's now been in many presentations. It's somehow lost pixels, I think, throughout each presentation, but you get the point. And so this was in stark contrast to if we looked at these same neurons in response to when the animal was consuming sucrose. So you can see that here in the group data. So this is the blue line aligned around sucrose consumption, red line aligned around shock, and you can see that there's a very different response here. So simple behavioral paradigm, but what allowed us to answer the question that yes, these neurons projecting from the medial prefrontal cortex into the periaqueductal gray are preferentially responsive to an aversive stimuli. And then we could go on to ask, does this pathway encode aversive outcomes that can occur during alcohol drinking behavior? So to do this, we needed to kind of design a new behavioral paradigm. So we needed this for a couple different reasons. One, for the conceptual questions we wanted to ask, but also to be able to combine this with the imaging. So a lot of what's been done in the field, in the alcohol field, is to just give animals access to a bottle with water and one with alcohol and let them drink freely. And of course, you can't really image across these really long time periods. So you kind of need to know when the animal is going to drink and have some kind of trial structure to align your data around. So the first goal of this paradigm that we were going to design was a very practical one, to include a trial structure that would allow us to integrate with the in vivo imaging. And then on a conceptual level, going back to what I brought up in the very beginning about these individual differences, we wanted to understand, be able to assess individual differences in both the consumption of alcohol and compulsive drinking, so along two different axes. And we really want to capture the longitudinal nature of the development of these types of behaviors. So again, what's also been done in the field normally is to give really long exposure times and then test compulsion at the end of that. And you kind of, occlude the individual differences by looking at after all the animals have consumed tons of alcohol, and you occlude the longitudinal development. So we wanted to assess this at baseline and then understand how it interacts with experience to ultimately produce these phenotypes. So this is what we came up with. Animals were conditioned in a chamber to associate an auditory tone with the delivery of sucrose to a port. So we used the sucrose just to train the tone, and as soon as they responded reliably, we then switched that out for alcohol. And then when we switch out for alcohol is day 1 up here. And so the animals get a fixed number of trials each day. So on each CS Plus trial, they can decide to either drink or not drink. And then we do that and give them access only to alcohol for the first 3 days. And then starting on days 4 and 5, we introduce graded concentrations of quinine into the alcohol solution. So we adulterate the alcohol with quinine. So quinine is a bitter tastant. The mice really don't like it. And so we use this as our measure of compulsion. So we're asking the animal, how much quinine are you willing to consume to still be able to get the alcohol? So here we're assessing, again, alcohol first and then compulsive drinking second. We then allow the animals to go through a binge drinking phase where they have free access in the home cage to alcohol or water for a certain period of time each day. And so using this paradigm, we can induce really high levels of consumption over days. So we're just trying to sort of induce plasticity and individual differences here. And then we come back to the original context that they were just punished, that they were last punished in. We reinstate them first for alcohol alone, and then go through this exact same paradigm of introducing graded concentrations of quinine to look for compulsive drinking. So this is what the data look like. You'll see a graph like this a couple different times. So again, to sort of reiterate what I said, you'll see that we hold the concentration of alcohol in the delivered solution constant at 15%, and then we change concentrations of quinine depending on the day. And so each one of these dots is an animal looking at their alcohol consumption connected over days. You can see the animals readily decrease when we introduce quinine here in the last 2 days. And then you can see again throughout the drinking throughout the binge phase, and then coming back here to the post-binge conditioning where we redo this same paradigm again. And so you can see there's a lot going on here, but one thing that you can hopefully appreciate right off the bat is that we really have a lot more variability over here in the post-binge conditioning than we do here in the pre-binge conditioning. And again, this is what we were going for. We wanted to see these individual differences come out so we can use that variability. So if you just focus here on the post-binge conditioning first, this is the data that we use to define the different phenotypes. So we took each animal's average alcohol intake throughout these first 3 days and plotted it as a normalized distribution on the y-axis down here. And then we took the average across all of the quinine days and plotted another normalized distribution across the group here on the x-axis. And so when we look at the data this way, we can see three distinct phenotypes come out. So we have animals that are above average in their intake of alcohol alone and above average in their intake of alcohol when quinine is present. And so we call those the compulsive drinkers. We have some drinkers that drink quite a bit, so they're above average in their alcohol intake, but they're very sensitive to the quinine punishment. They decrease right away. And so we call those the high drinkers. And then we have a group of animals that is below average on both of these measures, and we call those the non-compulsive animals. So we can then take this group definition that we're defining from the post-binge phase and apply it post-hoc to the entire data set, and then we can start to understand both the behavioral and neural phenotypes that interact with this binge drinking experience to ultimately produce this compulsive phenotype in the end. And so again, there's a lot going on here. We really put a lot of time into this behavioral paradigm, and it's something we're continuing to work on in the lab as well. So I'll just point out what's really important to know for the imaging data that I'm going to show you, but if people have questions about this at the end, we have a lot more data, and I'm happy to talk about it. So one thing that's really important going forward for the rest of this presentation, though, is that during these first 3 alcohol days here, the 3 different phenotypes are behaviorally indistinguishable. So they consume the same amount of alcohol during these first 3 days. Again, they're defined based on what they do over here in the post-binge conditioning. You can see even prior to the binge, there is some compulsive intake happening in the compulsive animals, although this is exacerbated by the binge drinking experience. But again, these first 3 days, the same intake between the 3 groups. And then we also calculated this vulnerability index for each animal. So this is just summing their value on the y-axis here and the x-axis here. And this just gives us a linear range that we can rank the animals on to correlate with our neural data later. So we were recording the activity of these MPFC to PAG neurons throughout this entire paradigm. So using the same approach that I said before with this projection-specific expression, implanted grid lens, and then an endoscopic scope throughout all of the conditioning sessions. So I'll show you the same type of representative trace here. This time, it's aligned around the initiation of a lick bout for alcohol, which will start when you see the blue square around the video. You can see our imaging got a little bit better between these 2 studies. So the animal starts licking here. And you can just see this kind of robust quieting across the field of view that sort of rebounds after the offset of the lick bout. So this was really interesting to see. You can see in this animal is really consistent across trials. So you have trials here in the heat map. This is just a whole field of view analysis where we take all of the pixels summed together. And you can see there's this really consistent inhibition of activity occurring around the time that the animal consumes alcohol. So when we break this down to the single cell level, so we used CNMFE to extract the different responses, again, aligned around the initiation of the lick bout here. And you can see that we see, even though as a whole we see this inhibitory response, you can see that there's this really robust heterogeneity in the different response types. So we use hierarchical clustering here to define it, and we actually see throughout 361 neurons, we get 8 different functional responses to alcohol. Quite a few of them here, as you can see, are inhibitory, but have different shapes and different dynamics after the offset of the lick bout. So this was pretty cool to see, but what really got interesting is when we started to break down between those 3 phenotypes that I described to you. So all the data that I'm going to show you right here is taken from the very first day of that whole behavioral paradigm. So again, at this time point, the animals are drinking the same amount of alcohol. This is prior to the binge, and they're behaviorally indistinguishable. So despite the fact that their behavior is the same, we can already see really robust differences in the neural activity. So these are heat maps now of single cells, not trials. This is averaged across all the trials in that first drinking day. And then if you want to just see the take home, just look at the trace at the bottom. So that's the average of the cells altogether, again, aligned around lick bout. And so you can see in the animals that are going to go on to become non-compulsive after the binge drinking, you see this sort of modest but prolonged excitation occurring around the time that they're licking. In the animals that are going to go on to become high drinkers, we see there's a lot of heterogeneity in the responses, but if you average it out, it's almost flat. And finally, in the animals that are going to go on to become compulsive, we see this robust inhibition of activity like I showed you in the representative video there. And you can see this come out once you put the cells into respective buckets as well. So in the non-compulsive animals, you have slightly more cells that are excited versus inhibited during the lick bout, and this decreases as you go through the phenotypes until you get down to the compulsive animals, which have almost 75% of the responsive cells show an inhibitory response during the consumption of alcohol. So we wanted to see how this correlated with the behavior. So we wanted to see, okay, it was very surprising to us that we were seeing these changes so early on, again, when the behavior wasn't different. So what we did was to calculate an excitation inhibition balance going on during the consumption of alcohol from, again, that very first day of data, and correlate it with each animal's vulnerability index calculated from the pre-binge behavioral data. So here we're looking at pre-binge neural responses on that first day versus the behavioral phenotypes prior to the binge as well. And when we do this, we see no relationship between the two. If we use the pre-binge neural data and try to predict the drinking during the binge itself, again, we see no relationship between the two. But if we take the pre-binge neural data and compare it to the post-binge behavior, we now see this relationship come out. So we can actually predict which animals are going to go on to become compulsive based on the neural encoding of alcohol during their very first alcohol experience. So again, we kind of hypothesized the directionality of this circuit with drinking behavior. So we thought, based on the shock and sucrose data, that decreases would be associated with more drinking. But it was really surprising to us to see this come out, so early on before the behavior was actually different. And so, based on these findings and the earlier imaging that I showed you, we proposed a working model that the MPFC is routing the valence of unconditioned stimuli downstream to the dorsal periaqueductal gray. So it seems to have inhibitory responses to stimuli that the animal then seeks out again in the future, and excitatory responses to stimuli that the animal is more likely to avoid in the future. And so if this working model is true, we should be able to test a few fairly straightforward hypotheses. So again, if this working model is true, we should see that the neural activity during alcohol consumption will predict future behavior on a trial-by-trial basis. So what I showed you so far is this sort of very gross correlation in behavior, but we should be able to see this come out trial to trial again if this is affecting the way that the valence of these unconditioned stimuli is being stored. And then activation or inhibition of this circuit should be able to produce unconditioned aversive or appetitive responses. And then perhaps most importantly, if we mimic the activity of these neurons that we see in these different phenotypes through activating or inhibiting them, we should be able to reproduce these drinking phenotypes throughout all of the animals. So we'll go through these one by one. So the first one, whether or not we can predict on a trial-by-trial basis. To do this, we turn to a machine learning approach. So this is using an SVM classifier. So we took all of the cells, again, this is from the first day here, and dumped them in together. We labeled each cell. So we just did binary labeling. So this is supervised. And all the cells, again, are aligned around this the lick bout for alcohol. And so we just use this labeling strategy. So it's slightly different than you may have seen people do it in the past, where they're using a cue and then labeling whether or not the animal goes on that trial. What we did was to actually take the alcohol response, the neural response to alcohol, and then label each one whether or not the animal consumed alcohol on the subsequent trial. So we're trying to say, does the activity in this pathway during the consumption of alcohol then affect the decision of whether or not to drink on the next opportunity to do so? So we did a bunch of statistical controls here to make sure there wasn't any issues. So we split everything into testing and training sets, and then iteratively permutated these to make sure that we were sampling evenly across the different phenotypes. Then we fit them, and then we create a distribution of our accuracy across 1,000 iterations of shuffling this training set and testing set. And of course, the testing set is never included in the training set here. And so when we shuffle all of our labels, we get an accuracy right around 50%, which we should. However, when we leave the labels on, we can actually see that we can predict with about a mean of 80% accuracy whether or not the animal will respond on the subsequent trial, again, based on the neural activity in these MPFC to PAG projectors during the consumption of alcohol. So this kind of confirmed what we saw with our correlational analyses and answered the question that yes, we were able to predict, at least to some degree, this future behavior based on the consumption during, or the activity during the consumption of alcohol. So then moving on to our second hypothesis here, we wanted to see if activation or inhibition of these neurons would produce unconditioned or aversive or repetitive responses. So to test this, we turned to optogenetics. So we injected pre-dependent vectors now carrying channel rhodopsin bilaterally into prefrontal cortex, and then used the same retrograde pre strategy into the dorsal paraqueductal gray to get projection-specific expression, and then implanted optic fibers over medial prefrontal cortex. And again, here we started with a very simple behavioral paradigm. So we just used real-time place preference. So for people that aren't familiar, the animal's put into a chamber. They're allowed to freely roam between the two sides. One side triggers a laser on period with no signal to the animal. The other side is laser off. And so on the laser on side, we just drove these neurons with a 20 hertz photostimulation. And so what you can see is that the channel rhodopsin animals, as compared to our EYFP controls, actually modestly avoid the laser paired side. So if we drive these neurons on 20 hertz, it appears to produce some avoidance behavior. And we also tested this, I won't go through all the data, but we tested this in multiple anxiety-like behavior assays and also looked at locomotion, and we saw no effects of stimulation on either anxiety-like behavior or gross locomotor behavior. So that data sort of matches with this idea that these neurons were responsive to the shock, and in drinking animals, when there was excitations, the animal were less likely to go back and drink again. So then we wanted to test the inverse, so we used a similar optogenetic approach, but now to inhibit these MPFC terminals in the dorsal PAG. And so here we use the same paradigm, but now the on side just turns the laser on continuously to inhibit the neurons anytime they're on that side. And you can see we get the opposite effect. So again, modest but significant effect where now the animals prefer the laser paired side when you're inhibiting these MPFC to PAG neurons. So we can say yes to the second hypothesis as well, that this activation or inhibition sort of matches the imaging in producing unconditioned aversive or repetitive responses. And so finally, the thing we really wanted to get to here is whether or not mimicking these endogenous activity patterns that we saw would allow us to reproduce these behavioral phenotypes as well in the drinking. So to do this, we use the same optogenetic approach. So I'll start with the inhibition here. So again, we're expressing halorhodopsin in these MPFC neurons and inhibiting the terminals in the dorsal PAG. And here we used a much simpler drinking paradigm. So we said, we had this long paradigm where we see all these individual differences. And we said, okay, well, if we're seeing these inhibitory responses coming out really early on in drinking behavior, before you'd really expect to see a lot of compulsion, can we actually just take that whole binge drinking experience out and produce these compulsive phenotypes just by reproducing that same activity pattern? So in this paradigm, we just gave a simple 2-bottle choice. So the animal goes into a chamber. Each session's only an hour long. They can lick freely on either a alcohol lichometer or a water lichometer. We do this over 3 days to establish a baseline. So again, this is a really minimal history of alcohol use, where you wouldn't expect to see compulsion. And then starting on day 4, we introduce graded concentrations of quinine into the alcohol bottle. And also starting on day 4, contacts on either the alcohol or the water lichometer trigger a 5-second laser on period. So we're only triggering the laser around the time that they're drinking, again, to try to recapitulate that inhibitory activity that we saw endogenously in the compulsive animals. And then in this paradigm, again, we just increase the concentration of quinine that's in the alcohol bottle over days. And so what we see, if we look at a percent of baseline, across the quinine doses. In the EYP control animals, we get a really robust decrease in alcohol consumption, even here at the lowest concentration of quinine, which is exactly what you'd expect for this very minimal alcohol exposure. But in the halorhodopsin animals, with just this 5-second inhibition, we can shift this curve upward and to the right. So we can more than triple the IC50 of quinine to decrease alcohol drinking behaviors just by inhibiting for 5 seconds around the time that the animal is consuming the solution. And importantly, like I mentioned, we also paired this laser on period with contacts onto the water lichometer. And we see no change in water intake here. So this seems to be specific to this compulsive alcohol drinking phenotype. So we also wanted to test the inverse, whether or not we could decrease these drinking behaviors. So instead of testing the exact inverse and seeing if we could augment the effects of quinine, we said, well, if we are hypothesizing that these neurons are encoding, that excitatory responses in these neurons are encoding some kind of negative valence for an unconditioned stimulus, maybe we could just remove the quinine entirely and decrease drinking behaviors just by reproducing that activity pattern. So again, we use the same behavioral paradigm. Now the animals are expressing channel rhodopsin in these MPFC to PAG neurons. After 3 days of baseline, we don't put anything in either the alcohol or the water bottles, or no quinine. You know, they still have alcohol and water. But contacts on either lichometer now trigger a 5-second 20 hertz excitation. And there's no quinine here again, so instead of changing the concentration of quinine, we just increase the light power with which we're recruiting this population of neurons over the test days. And so what you can see is that the effect is much less robust than quinine itself, where we can actually extinguish all the way, but we can produce this light power dependent decrease in licking for alcohol, again, outside the context of quinine entirely, just by pairing a 5-second 20 hertz excitation with contacts onto that lichometer. And interestingly, in this experiment as well, we included a recovery period afterwards where we allowed the animals to keep drinking in the same type of session over days, but did no manipulation at all. And you can see that pairing the stimulation with the consumption of alcohol actually has a lasting effect on alcohol consumption that recovers over 5 days. And this is exactly what you'd expect to see if you punished alcohol drinking with quinine or foot shock or some type of normal punishment as well. So we think that by reproducing this excitatory pattern, we can actually partially recapitulate the behavioral effects of punishment on alcohol drinking behavior. So just to summarize everything that I've shown you, we showed that these MPFC to dorsal PAG neurons bidirectionally signal the valence of unconditioned stimuli. So it seems to be excited to aversive outcomes like shock, inhibited or nothing to appetitive outcomes like sucrose. We then developed this novel compulsive drinking paradigm that allows us to track the development of these behaviors across long periods. And we show that the activity in these neurons during the very first time that the animal consumes alcohol is actually predictive of whether or not they'll go on to become compulsive drinkers more than 3 weeks after this recording is taken. And finally, by recapitulating these signaling patterns, we show that we can drive compulsive drinking with the inhibition of this pathway, even in animals that have never had this binge drinking experience. And we show that stimulation of these neurons is sufficient to act as a punishment and decrease alcohol drinking. So I'm going to end pretty early here to give time for questions and everything. So I just want to really quickly talk about where we're going with things in the future now in my own lab. So this is some more recent imaging. This one's a little more post-processed and sped up, so we're cheating a little bit. But you can see, once you optimize these, that you can get some really, really nice imaging in freely moving animals. But one of the things we're most excited about with these data is actually the ability to spatially follow these neurons over long periods of time. So if you look at this recording just as a T projection, this is the first time we recorded from the animal. 2 months later, 4 months later, 6 months later. And so, this is actually a little Inscopics history. I don't know how many people of you have used these before, but the old Inscopic scopes, we actually had to manually turn the turret to focus them. So you can see a little bit of rotational change here, but it's pretty obvious that we're getting these same constellations of neurons. And now we're doing recordings with the newer scopes that have the electrolytic lenses where you can just focus without turning anything. So we get rid of that too. But you can really clearly see these same neurons over time. And this is really exciting because, especially in something like alcohol use disorder where it's a really long development, we've never been able to follow neurons in vivo throughout as long as we would need to understand, is it the same neurons that come online over drug experience that cause changes in behavior? Is it different neurons that are recruited to ensembles? And so here we're actually now collecting data sets where we're basically observing the activity of the same defined neurons through literally every single time that an animal interacts with alcohol or an alcohol-associated context throughout their entire lives, throughout their entire adult lives. And so we're co-registering these over time, and the other thing we're working on is also co-registering across imaging modalities. So we've taken these same animals, same grid lenses, attached a head bar, and put them under tabletop 2-photon microscope, where we can do head fixed behavior. This is just one of the earlier examples, but you can see this is through the same grid lens, so you have to squint a little bit more on this one. But you can see that same blood vessel down here in the bottom right, and some of these same constellations of neurons come up. And so now this is sort of a work in progress, but we're now trying to co-register across these imaging modalities so that we can actually collect all of the endogenous activity during free behavior and then come onto a tabletop microscope and do all sorts of, all the different manipulation approaches that are afforded to you there. So we can do structural imaging, we can put in different opsins and things like that to cause plasticity under the 2-photon scope and then come back to the freely moving scenario. And so, I talked a lot about the alcohol and drug abuse stuff today too, but we're really think that these sort of technical advances are going to open a lot of doors for many different questions. So we're also exploring a lot of basic learning processes, and being able to follow these neurons throughout learning in many different contexts, I think is really going to open a lot of new directions for us. So with that, I would like to thank everybody who was involved in this. So of course, like I said, almost all the data that I showed you was collected at MIT when I was a postdoc in Kay Tai's lab. So huge thanks to her and everyone in the lab that contributed. So Caitlin Vanderwill, we worked together a lot on the initial recordings in the shock and sucrose paradigm that I showed. And then for all of the alcohol work, I'd especially like to thank Habiba Nomani, CJ Chang, and everyone else here. Alex Brown also, who was in Kay's lab and now is in my lab, also contributed a lot to this work. And then everyone who's with me now at Vanderbilt. So with that, I'll do my best to answer any questions.
Thank you, Cody.